All right, welcome back to the last session before break for lunch in the food trucks out there. Looks like the fog has cleared, so we get to enjoy that pretty good. And with that, I'd like to welcome Dan and Basker to the stage to share their stories with Elasticsearch and how they've been integrated with human genetic data. Let's give them a round of applause.
Hello, I'm Dan, here to just give a little introduction to our journey of using Elastic and helping drive drug discovery. So here's our agenda for today. Let's do a brief intro. We're going to go a bit into the science of what's going on, because we're a little different from what you guys have seen so far in other talks. We're not logs. We're not time series. We are genetic data, and it's a little different. So we are Merck. We are a global drug company. Outside the U.S., we are known as MSD. We produce a variety of drugs and vaccines, both large and small molecule, and this is what we do. So who are we? We are engineers in the scientific computing group for the Merck Research Labs in the early discoveries portion of the drug discovery process. When making a drug, there's a sort of whole suite of activity being done on the chemistry for making a compound that'll work on a certain target. On genetics, in the group that we support, is about, is there genetic evidence to support that this drug will actually do what it's supposed to do, and that's the basis of our talk. And there are other part of, does the drug work in the body? Does it go through your blood and the liver, and does it break down correctly? So there's a lot of activity going on to prove safety and efficacy that needs to be done before it's even put into humans at all. And for us, we engineer and build stuff that may or may not exist from a technical perspective. So what is the billion-dollar question here is that as an industry, in the pharmaceutical industry, in the early discovery process or even in phase 3, there is a pretty low batting average for what we as an industry are doing to produce drugs, and if they are indeed effective and safe and cost-effective. Can we do better? And we think we can with the human genome, being completed about 14, 15 years ago, this wealth of genetic data coming in. We think we can do better. And studies have been showing retrospectively that, looking at prior drugs, you can see genetic evidence being a predictor of probability of success beyond phase 3 that will indeed be valuable to mine the latent genetic data that's coming in and see if we can use that as an additional probability of success. And one argument is that we have, as an industry, we have cured every disease in mice, but that hasn't necessarily translated in humans. So the summary here is, get the wealth of genetic data, mine it, and hopefully we can then improve probability of success, maybe reduce the misfires, and get drugs to market sooner, safer, and hopefully cheaper. And so here's the global approach here. It's a big village that is building this. We are in this area of the process. Genetics and pharmacogenomics, again, is the group that we're supporting. And in the end, our problem here is a find and filter problem. There's a lot of data out there, and how do we make the scientist's life easier? And so, before we go on, let's do a little science lesson here. Definitions first. Genomics is structure, identifying what our genes are, what the nucleotides are. We know that nucleotides encode the proteins. Genetics is more what I have. It's an instance of my code and what is heritable. Genotype is the code again of what is in me if I get myself sequenced. And phenotype is the observable characteristics, black hair, brown eyes, or skin color. So, and the core sort of unit we're talking about here from here on forward is the variant, or the single nucleotide polymorphism. That is the actual diff from the reference of your genome, your A's, G's, and C's and T's, in what I have in my genotype. And so it actually is fairly common in your body for everyone here, and that sort of defines what is different among all of us. So code, that's interesting. We have a lot of engineers here, so let's dive deeper here. So every few years, we make a reference genome, and it's sort of like the kernel, Linux kernel, and we say, okay, 23 chromosomes are changed, and we have 3.2 megabase pairs changed, so 3.2 billion, a million documents. So let's check myself out. Let's see who I am, and then let's do a merge because I'm born. And now let's do some magical unit test to see what's in me, what is happening in me. And I see, okay, I don't have asthma, I am lactose intolerant, and I have some indicators for cholesterol here. And so what could I do? I can just go find out who gave this to me, and then, oh, okay, thanks. And so, one thought is you can ask your parents. They might just say, oh, works on my machine. But really, this is still also an interesting place where, heritable diseases, cancer susceptibility, this is pretty important history to figure out as well, just not only for jokes, but for, understanding how things are actually happening in our bodies, and hopefully find therapies there. And then from there, let's find out the diff again, and we have all these minor changes here going on, and these are SNPs, or these are the variants going on. So why are SNPs important? I just did an example for myself, but why do we need to get as much data as we can? Because in my single instance, I have some indicator of some susceptibilities here and there, but in a population across thousands of people, these variations form a pattern or will try to form a pattern. And now what's happening now is there are a battery of studies going on to sort of figure out if we all get sequenced here and sort of submit, and get our SNPs figured out, then there'll be all minor changes of hundreds or thousands or millions of variants here that may point to diseases or susceptibilities. In actuality, having variants, a lot of them don't do anything. So there's a lot of noise that we need to filter out. And so now we understand sort of the basis of our unit. Why is this important? Let's talk about the weather. We have 10,000 years of civilization that have weather, and we figured out how to do that with latitude and longitude as our basis of a location. And now we have synonyms for location with addresses and regions and state boundaries. And we have sensors and observations overlaid on top of these, and other things like check-ins and trips and maps and routes. And from there, we can start making predictions on top of these things, thanks to having a location system like latitude, longitude, and altitude from there. For us, in our genetics world, we have 13 years of figuring out what is in our DNA. And so we have finally chromosome and position we can base ourselves upon. And then we have synonyms with what these regions and locations are, with gene and variant. But then after that, we have a series of observations that we overlay on top of that. So gene expression, epigenetics, and then linguistic equilibrium, and other bits of expression and activation that are happening based upon what variants you have. These are still in their early form of how to quantify these things, but these are all part of the activity that's going on to understand the causes of disease. And then from there, we are hoping to have those predictions and insights just like the weather. Right now, for our basis of our work, we're still over here. I mean, our goal is here, but we need to get stuff into Elastic first. So that's why we're talking. So recap again, the need. It's wild out there. We have 13 years of rapidly declining cost of sequencing. The standards in which the data is being produced changes every few years. It's tedious, and if you read this book, the first 2/3 of the book for a bioinformaticist is learning FTP and just getting these data from these public data sources and just cutting, auditing, and producing a CSV file. And then finally, it's slow. We've had our own sort of issues with dealing with the scale and the variety within the work we're doing, and answering some basic questions will still take hours, weeks, or months, depending on the type of the work that we're doing, due to the variety that we're dealing with. And so, why are we here? There are a number of case studies around that inspired us, and the first one being actually ElasticCon 2015. The Yale came and spoke about their variant database that to search for patients' genotypes to help them enroll them into clinical trials. That set the gears running in our heads, say, oh, we can do this too. But there are sort of three dimensions in which we have a need here. There's variant annotations, which is just identification of what is out there. There's phenotype association, which is the diseases, observed diseases, and sort of the observed activity. And then the actual other data, such as GWAS and EQTL, which are the meat of understanding cause and effect. So there are other industries out there. So out of Scripps in San Diego, MyGene and MyVariant is another sort of lookup service that really helps you understand what is out there for genes and variants, annotating all the different synonyms around there. And then for Open Targets, which is a multi-pharma industry consortium about linking variants to diseases to say, oh, HMGCR is related to heart disease, or PCSK9 as well. And then type 2 diabetes genetics is another portal that tries to sort of link those two pieces together with the GWAS and EQTL studies that are coming out now to start mining genes to disease and the underlying data for that. And our desire is to be in that sweet spot, but more than diabetes, for across a broad area of diseases here. Turns out everyone is almost using Elasticsearch, so why not? Let's join the fun. So Bhaskar will take over.
Hello, everyone. So far, biology enough. So now we are engineers. How the process of getting into the data into Elasticsearch. I'll walk through the journey of in Elasticsearch from data cleaning to visualizing the data. Here the goal is we need to map all the public genome-wide association data into the single unified identifier that we are calling as a SNP. And those are all the different observations into like a parent-child document here to our variant doc. So same kind of what Dan mentioned of the analogy of maps. Here we can take about the SNP as the geolocation and the observations of the EQTL and GWAS there. And this is additional guidelines we are following for normalizing every data study from the GWAS multiple studies into keep the common nomenclature. Just if you have the same fields, just keep it into the same nomenclature. If anything, study-specific fields, just keep it as the X. The single data harmonization, I think if you give it to the bioinformatics, the team, they can talk about 2 weeks on this single topic actually. This is our secret soup of what we are doing here. This is our document structure. Variant, as like the same thing, geolocation on the whole genome wide. That's our parent. And all the observations, GWAS, PWAS, EQTL, frequency, genotype, expression data, these are all childs. So one variant have the 100 to 200 childs for each document type. So this is our variant. This is our ID, uniquely identifier across all the different studies. So now that's B37 is the genome version, chromosome position, all that's specific to the SNP. Doc examples from the genotype, one parent, you have the different childs over there. So you can see this may have 3,000 to 5,000 for each variant. So I just showed the small example here. This is going to be wide. And the same way, GWAS, one variant may have the 100 to 200 childs. Same thing, EQTL, the same thing. These are all like a expression and the genome-wide association data. So what is the allele frequency? Allele frequency is like a frequency on a particular variant for a given population. So it's like in same thing. For a given variant, you may have the 100 population, subpopulation data. How the mapping we started with Elasticsearch 2.4. Based on the nature of the data, so we kept everything as a nested. So one document with 100 to 200 with a nested document. It's good, slow indexing, but faster queries. But the day of the natural way is consuming is good, but its data keeps updating. So when we add a new document to the existing parent, it's so expensive. So then we choose the parent-child thing as the next version. It's good. Loading is good, but we need to pay for the as parent as child queries. They are really slow. So right now we are following the two-step query process. Get all the parents and go to the child and the child queries basically. It's a two-step process. Then version 3.4, we chose if you're not querying, don't index. Just index, make it as a false. So the first two versions, we used to have like a 10 terabytes of data. Then we make it to the 1 terabyte because I showed the genotype. There's so much big array of data sets there. So we kept it as to index false. So that's sent from 10 terabyte to the 1 terabyte. Version 3.4, this is current what we are doing is upgraded to ES 5.1.1. Still, we are following the parent-child schema structure. Okay, performance is okay, but we are still facing the performance problem because we are not doing the aggregations or something. We are just filtering the data. We want each and every point for a given query. So it's sometimes more than 10,000, 70,000 points too. This is our structure for a given one variant. As I mentioned, frequency, EQTL, GWAS, all the different stuff. So far, this is our usage story. Users want to see each and every data point. That's like 10K plus, more than that. They want to see everything in the given complete structure. We are in the edge right now. We are using scroll. That's where all the ADA queries. Which we are not indexing. We are using the painless scripting to all the linkage equilibrium referenced by LD queries there. So sometimes pairwise calculations too. This is our data loading. Data comes from public sources, public consortiums, and the academia. GTEx portal, exact, there's so many. It's like 200 to 300 different sources. The first thing, as I said, harmonizing the data, because the analogy is everybody use their own versioning and everything. So we need to make it as a single nomenclature. That's harmonizing and make it to the coordinates to the single magazine. Then use the Celery to parallelizing the process, and we are keeping to the Elasticsearch. This is our topology using 12 data nodes, 3 master nodes, applying XPack because each data set is a secured, so we are doing at the document level security. Django for the REST APIs we are creating on top of the Elasticsearch, we get REST APIs. Postgres is just for the storing users and and Kibana is for managementing and the logs, who is using what. So Aurelia is a JavaScript framework. Plotly we are not using definitely Kibana for what we are visualizing. We are using Plotly, based on nature of the graphs what we have. Jupyter for advanced users, or also for advanced analytical purpose right now. This is current stats what we have in our thing. Total, this is across genome wide, each variant thing. So total variants, we have 100 million parents. That's 100 million variants there. These are our childs. Currently, these are like 1 billion or something, but this is going to grow by 2 to 3 times in just a year. So it's like we have just got 20 to 30 studies. We have so many studies out there, 200 to 300 studies. So we have our own internal website we created for this. So you can search by gene, variant, region, and you can explore what are the variant existing for a given study or everything. And you can even explore for a given phenotype how that across globally all the chromosomes level. So this is just like give the description of what we are getting from that one. This is our beautiful chart there. So now this is given study across 24 chromosomes. So we can see where is the peak over here. So this is where scientists can focus, and is it really study they can focus or not from here. GWAS, this is genome-wide association. X-axis is this is x-axis for a given chromosome. What is the length of? We are search for HMGCR. This is our gene search, and we are doing the plus or minus 250 kb kilobase base pair of left-hand side, right-hand side. You see, search for gene, but there are other genes in the left-hand side, right-hand side, and you can see for a given variant which diseases are associated here. This is the topic you can focus on here. It's a lipid measurement for a given HMGCR, and we are not scientists or something, so we are engineers. So needs an expert to explore even this chart. Dan can take out from here.
Yeah, so we've been sort of talking about HMGCR a couple times. HMGCR is the gene related to many of the statins that we are familiar with for cholesterol-lowering drugs. It is sort of the prime example for the retrospective look at why genetic evidence is important, that the gene lipid count, in the neighborhood of HMGCR, it's showing an interesting effect there, and it is the starting point for looking into, is this another viable candidate for a drug? Need expert interpretation, plenty of opportunity for spurious correlations. So correlation is not causation here too. So it just presents in a way that previously would take weeks to hours across a broad array of studies to start asking questions and start testing hypotheses. EQTL is another type of a graph here about a variant and what it does around the gene there to see if you're having a variant will turn off or turn on genes in the neighborhood of and other genes in the neighborhood of that chromosome that you're on. So it also presents questions about if that activity is happening, are you different than people who are homozygous for the normal variant, or if you're heterozygous or homozygous with the actual variant there, does it actually turn on or turn off other activity to once again start finding hotspots to start asking more questions there. So needs without saying, consult an expert once you see something. So conclusion here, this is a start. This is a start. We put something in one database so you can search things now. And from there, we want to embark on additional looks into that or find other ways to help facilitate the search for causal activity and other correlations there. So the next steps would include sort of trying to applying machine learning, or right now even basic statistics on this broader data set is sort of first step there. We are looking at seeing if Elastic can assist in some of the pairwise calculation instead of doing a lot of data pull out and running it in R and then putting it back in. That's a lot of data coming out just to do one type of calculation. We've looked into that as well, and using other facets as well instead of pulling so much data out. So that's where we are right now. And thank you. Any questions?
Awesome job. Thank you, Dan and Bhaskar. Any questions for them? Okay, we'll go in the back, and then you next.
Question to Bhaskar. So when you talk about ingesting the data into Elasticsearch, so what kind of tools you use, or what process you use to ingest the data?
So right now we are using Python Celery, so
Okay.
parallelizing with the RabbitMQ messaging.
Okay.
And we are doing like 17 to 18 parallelization on top of that one.
Okay.
So
So how much is the volume you update in one or index in one shot?
this is, we are keeping everything in the single index. It's a static
Yeah.
data.
Yeah.
So right now that probably 17,000 per second or something, but that again depends on the number of nodes.
Right.
So-
So we do the same thing, like when we went from parent-child to denormalize and then use nested objects to store our data. So for that we use Logstash to update the thing. So it may be helpful. I don't know.
Yeah, that we heard from the stocks. Actually, probably we're going to think about it because that's where we can increase the volume of
Correct.
loading too.
Yeah.
Yeah, that's what we are looking to.
Okay.
That's what we did.
Hi, I'm with a group at Stanford, and we're actually part of the ClinGen project that's like evaluating data. So if you need experts, we probably have those contacts. But we are a publicly funded, grant-funded organization. Do you guys share any of the data, or will you expose any of the data via APIs? I imagine a lot of the data that you want to keep is proprietary, but are you going to share that?
I'll have to get back to you on that. Many of these efforts, including open targets and type 2 diabetes, are seen as pre-competitive. So most likely, at some point, it will be public, just the raw data, but it depends on the producers of the data. Obviously, these are all de-identified sources of the aggregate data, but most of the efforts going on, these consortiums, have been multi-pharma efforts to raise the tide for everyone. So that is the trend that we've been observing as well from the industry.
I think we have one more over here, and then.
Hi, I'm Ankit from Lumada. When we are in healthcare, we have the same kind of a data model. Oh, hi, I'm Ankit from Lumada. We are from healthcare. We have the same kind of data model. I want to know why you chose child-parent data modeling over nested docs.
So the nature of the data, so think about from the GWAS thing, so it doesn't come at once actually. So each and every study comes like monthly or something. So we want to update into the nested thing. That's like taking a long time, and it's like a very expensive process too.
If one variant had 1,000 GWAS inside it,
so then you need 2,000. Yeah.
We have 20 other GWAS studies come in, update that one variant. We'd have to pull that document out and update and all that other stuff. So
it's like expensive.
So the child docs that I saw, the payload is very small. So it's not really big payload to insert one parent child with having 20,000 child, or 30,000.
Oh, for the query costs?
Yeah, for the insertion. Because with child parent, querying is really slow. Aggregations, you don't get much out of it. So you cannot get inside if you cannot do aggregations on such data models.
I think aggregations are fast. I think we see the parent-child aggregations we didn't have any issues, but
yeah, we haven't crossed the parents with aggregation counts that yet. We just only at the child level aggregation or at the parent level. We haven't really done,
for a given parent
bucketized by the counts or something like a p-value count of a GWAS count for these type of things. We haven't done that yet. It's probably we haven't seen it try that yet.
Okay. Yeah.
Are you loading individual subject genotypes or study evidence?
Study evidence, yes. Subject genotype,
no.
For right now we're just doing public level data, so they will have the calls in them.
And you're loading those calls?
Yes, but not indexing. We're just literally loading it. I mean, it goes back to the go to the flat file. Oh no, where do I find it? So it was a nice place to store it. And then some of the calculations we could do, we have it conveniently in the document, and then we could run the one that we're doing right now is LD calculation. So just get all the genotype data out with the population and then do the math on that.
Probably we're going to think about painless scripting on top of that one too. So.
Any other questions? All right, let's give him a hand.